Hi everybody, my name's Nathan. I'm an MD PhD candidate in the Sioux Laboratory in the Center on Aging at UConn Health, and I'm here today to talk to you about some of the exciting opportunities in the field of aging research. Aging research is a critically important field with profound implications for both healthcare and society as a whole. Over 1 in 6 Americans are above the age of 65, and as you can see from this figure on the lower left from the US Census Bureau, that number is projected to increase significantly over the coming decades. And this is of particular concern, especially given that age is a major risk factor for the leading causes of disease and death, such as cardiovascular disease seen here, as well as death from dementia, pulmonary disease, infectious disease, kidney disease, and more. Therefore, one of the current aims of the field is to better understand what it fundamentally means to age so that we can improve our treatments of these diseases and potentially even correct some of the changes that occur with aging. And altogether, this might allow us to live longer and healthier lives. So in our lab, we primarily study aging using a mouse model system, and we have a number of reasons for using mice. In particular, mice are mammals just like us, and we also share a great deal of genetic similarity. Furthermore, mice only live about 2 years on average, which is a lot more convenient to study in lab. Additionally, there are a number of genetically modified mice and other experimental systems that allow us to ask questions about specific aging-related diseases, such as Alzheimer's dementia or kidney disease. Our group uses a number of techniques to study some of the physical changes that occur with aging. For instance, we can place mice on a miniature treadmill to determine how fast or long they can run. We can also test their grip strength, or even give a complete physical exam to assess for changes such as loss of body weight, graying of hair, loss of hair, or changes in overall body condition. We also perform a number of behavioral tests, particularly in our mice with cognitive dysfunction, such as Alzheimer's models. In particular, we can assess for changes in sleep-wake cycles, changes in their eating behaviors, and even changes in memory. One example of a memory test is this Y maze shown below. So during the first trial, a mouse is placed into this maze with one arm of the maze closed. Behind that arm is something of interest. For example here, a piece of cheese. We can then wait a number of hours and place the mice back into that maze without that object or stimulus to see if they remember where it was by measuring the amount of time they spend in that arm of the maze versus all other arms. Additionally, we assess organ function in our animals using a number of molecular techniques in microscopy. In particular, we can look at circulating proteins, we can also assess liver, kidney, and other organ function, and further assess cellular aging markers. Our lab is particularly interested in a process known as cellular senescence, a cell fate that increases with aging and disease. Senescent cells are cells that stop dividing and produce a characteristic set of inflammatory proteins. Unfortunately, there's no great single gold standard marker for senescence, and we instead have to rely on multiple experiments to determine if these cells are there. One particular assay I'll talk to you about today is called the telomere-associated foci, or TAF assay. This system measures persistent DNA damage signatures that accumulate with aging, specifically at the end of chromosomes, known as telomere regions. First, we transfer a tissue of interest to a glass microscope slide, similar to one that you might have seen in lab. Here I'm showing an example of skin tissue. Next, we place our samples into a slide rack in order to perform a number of staining and washing steps. We can use fluorescently conjugated antibodies to label our DNA damage marker of interest, as well as fluorescent DNA probes to label our telomeric region. So after some additional washes and incubations, we're now ready to image the slides on our lab's fluorescence microscope. Here I can be seen placing some of those stained slides into our microscope's slide holder. This holder is then placed into the microscope, which is controlled via our computer system. Finally, we take a number of images throughout the slide to quantify the areas where we have staining for both our telomere as well as our DNA damage marker and find where they overlap. Here are a few representative images from our experiment. In green we have our DNA damage marker, in red our telomeres, and our cell nucleus is labeled in blue. As you can see on the left, we have a lot more of this telomeric DNA damage in our diseased tissue versus our healthy tissue on the right. On the far right, you can see we've quantified this for a number of samples. So I'd like to wrap things up by talking about some of the exciting careers you can have in science. In particular, I'd like to talk about my path as an MD PhD student. An MD PhD provides training through completion of both medical school and graduate school. After college, students will typically complete their first 2 years of medical school before going on to their PhD training. After this, they'll complete their final 2 years of medical school and go on to residency and postdoctoral training. While there's definitely a lot of school involved, it can certainly be a rewarding career. For instance, MD PhDs can work both in a clinical setting as well as lead a laboratory. In addition to the direct ways you might improve people's lives in clinic, your research in laboratory might help you provide new cures and treatments where there aren't currently options. In addition, you'll have a lot of great career flexibility. For instance, while you might work in an academic teaching hospital, you could also work in the scientific industry. If this sounds interesting to you, UConn provides MD-PhD training right here at UConn Health in Farmington. If you have any questions about aging research at UConn Health or MD-PhD training, please reach out and I'm happy to answer any questions.